welcome to this webinar, From Patient to Petri Dish, Replicating Disease with Patient-Derived Organoids. I'm Diana Spencer, Senior Digital Content Editor at DDW. Developing new drugs is a complex and often lengthy process, limited by the fact that traditional preclinical models do not always accurately represent the intricate biology of human patients. In this webinar, Dr. Sylvia F. Boch, Chief Scientific Officer of Hub Organoids, will explore how Hub Organoids faithfully recapitulate patient-specific pathobiology, remain genetically stable over passages, and readily incorporate genetic modifications. She will explain how this combination bridges the gap between the lab and the clinic, enabling the development of more effective therapies. In this webinar, you will learn how Hub Organoids overcome limitations of traditional models, providing a unique solution for disease modeling and drug development. The latest innovations in organoid model development, including co-culture systems, expansions to diverse epithelial origins, and customized assays tailored to the specific research hypothesis. And the real-world success stories showcasing the impact of Hub Organoids across various R&D projects spanning immuno-oncology, inflammation, infectious disease, and personalized medicine. I'll now hand over to Sylvia.
Thank you very much for the nice introduction, and thanks to the audience for joining today this webinar. The title of the presentation is From Patient to Petri Dish, Replicating Disease with Patient-Derived Organoids, and I'll take the next hour to introduce our technology, patient-derived organoids, and how we are supporting drug development pipelines. Hopefully the audience is familiar with the traditional process for drug development, that it's quite inefficient since we start using not very relevant preclinical models in order to identify compounds that are moved towards clinical phases. We are quite aware of how expensive and how much time takes to take drugs into the registration phase, and still a very low percentage of compounds and drugs that reach this funnel end up on a clinical on the treatment of patients. We are talking about between 10 and 50% of the drugs. So we believe that we can bring value and provide a solution to this challenge by using patient-derived organoids on your drug development pipeline. And the earlier you do it, we are convinced that more efficient in terms of cost and time, the development of your compound that really will have an impact on patients will benefit. And we just want to bring this idea of having more centralized patient approach in order to develop drugs. So what are these patient-derived organoids? So these are in vitro models that in our case, and based on our technology, they are derived by adult stem cells, as it was shown for the first time by Toshiro Sato with intestinal mouse organoids. So I also want to emphasize that in this sense, we are not using the iPS technology. It's based on these LGR5 positive cells identified in almost all intestinal epithelium. And then with this technology, we can develop 3D in vitro models that are physiologically and biologically relevant. They represent the characteristics of the tissue and the cell composition present on each specific tissue. The efficiency of establishing this model is very high if we compare it with the establishment of other preclinical traditional models like cell lines or PDX. They are genetically and phenotypically stable. The culture conditions do not induce any transformation of the cells. They preserve patient and disease-specific antigens, and we are taking advantage of this to have more human-relevant models in the bench. The good thing is that they are amenable to co-culture with immune and many other cell types, so we can also develop as many complex assays as we need, depending on the question that we want to answer and the challenge that we want to address. They can also be they can be generated actually not only from human patients but from any animal model available. Especially we can have a collection of preclinical animal models represented on organoids. They are expandable for large scale screens. They are suitable for genetic manipulations, and of course they are because we can establish organoids not only from diseased tissue like inflammatory diseases, oncology, but also from normal tissue. We can have the normal counterpart of any diseased tissue in parallel on the assays. It allows us to also evaluate on-target or off-target toxicities effect. But one of the additional and more valuable characteristics of these patient-derived organoids is the predictive value of these organoids to patient response. And this is as an example of one of the first cancer biobanks developed in the past. In this case, based on this is the biobank for breast cancer, in which out of biopsies the organoids can be established. They can be characterized at histological molecular level, also evaluating the sensitivity to compounds, but very importantly, because we have clinical information from the patients from which the organoids were derived, we can also compare and evaluate if the organoids are predicting the response that the patients have in the clinic. And this is one example of, in the context, as I mentioned, of breast cancer, in which we could establish organoids from two different patients. The patient in pink, the organoids derived from the patient in pink was a patient that was showing progressive disease in the clinic, so the patient was not responding to treatment, while the organoids derived from this more greenish, bluish organoid was generated from a patient that had a response in the clinic. And actually, we could see that the organoids show a different sensitivity to the compound, the same compound used in the clinic, where the organoids from the patient that was responding clinically were showing more sensitivity to the compound than the organoids generated from the patient that was not showing response in the clinic. We see the same trend in other type of cancers, like this metastatic colorectal cancer, in which we are running a clinical study with our collaborators at UMCU Utrecht, in which we are now collecting biopsies from patients that are diagnosed with metastatic colorectal cancer lesions in the liver. The patients, after the isolation of the biopsy, start receiving treatment in the clinic for a period of months. In parallel, when we get the biopsy, we establish the organoid cultures and we run the sensitivity of the organoids to the specific treatments, the standard of care now in metastatic colorectal cancer. And what we are seeing, and this is just a subset of 20 patients, this is a study that is aiming to analyze more than 100 patients, is that we see a very significant correlation between the response of the organoid, in this case evaluated here as an area under the curve of the organoid area when exposed to the compounds, with the clinical response that in this case is the size change in the lesion of the patient. So in the size of the graph means that the lesion is reducing, so the patient is responding in the clinic, and here the area of the organoids is smaller, meaning that the organoids are responding to the clinic. So with all this combination of information about the genetic and biological and cellular complexity that the organoids bring in terms of patient representation, but also the patient response, we believe that patient-derived organoids are more relevant platform to develop new drugs. Actually, we are quite excited to share with this audience that quite recently, 2 weeks ago, we received the award from Frost and Sullivan about our technology by the high impact that the organoid technology can have on drug development. So we are quite excited to what we've been able to develop already and prove the value of our technology, and looking forward to keep expanding the utility of this technology. So in this graph, we tried to kind of summarize very high level what is the process of drug development from target discovery to running the actual clinical trials with patients to prove that the compound has an effect in patients and can be used for the treatment of certain diseases. And actually, we see that currently organoids already have a relevant positioning in the area of lead identification, lead optimization. So we already have developed a number of assays and represented a number of diseases with different type of organoids, in which compounds in this area are already kind of being tested or evaluated before moving into clinical trials. But actually, we have the ambition to extend the use of the technology in early stages of drug development, because we believe that when the earlier that organoids are included in the pipeline, higher chances to select the right compound towards lead with a relevant, clinically relevant impact will be identified. And we also see the value of the technology really moving towards patient stratification and co-clinical trials to have better patient stratification for clinical studies, so to understand immediately have more understanding for the group of patients that are not responding on the clinical studies. So with these efforts of expanding our technology across the drug development pipeline, we have here a summary of our services, our offerings, in which we have what we will call drug screening services, in which we can perform very straightforward assays with very clear readouts in a define a collection of organoids to test the activity of certain compounds with multiple readouts. But also we have additional services like this custom model and assay development, in which we have a common interest together with our customers to expand the use of the technology in different diseases or in different models, developing new assays. But also we see the value again of running co-clinical drug developments, where we can generate this organoid data in parallel on the execution of a clinical trial to gain more insights about what is happening with the patients, and have, for example, a population of samples from which, if the patients are not responding in the clinic, we can already do some studies to understand why this group of patients are not responding. And also our technology can be out licensed, so there is the possibility to license our technology and work in-house to implement this platform for your R&D and drug development pipelines. And then that we think it's a great combination with our service offerings in order to accelerate some of the implementation or some of the progress of your drug discovery pipelines or projects. So in this slide, we summarize a bit where our main expertise. From one side, about the disease areas, there are already some of them where we have both organoid models that represent patients for these specific therapeutic areas, but also relevant assays in which we can evaluate the activity of certain modalities of compounds. And also, very importantly, this is the collection of tissues from which we can develop organoid models, all of them human-derived models, but we also have animal collections for specific organs and tissues to explore how indeed our technology could support the toxicology area. So actually on today's presentation, I'll focus on mainly talking about inflammatory diseases and toxicology as two potential areas where we can where we are going to show some of the developments that we have performed the past years. So actually on this model assay development services, what we try is to first, well, define if there is a need for a specific disease or for a specific compound, which mechanism of action requires an assay and an organoid model that is not available now in our portfolio, so that we can develop it together. We can agree on what are the relevant endpoints for the assay, what are the readouts of the assay that will make more sense from the patient response perspective, from the clinical perspective. Work on the assay development either by fine-tuning the conditions where the organoids are expand or the assay is performed, and then we start kind of developing the assay and generating the data to prove that the assay or the biobank that we are generating fulfills the requirements or the criteria needed to get it kind of adopted and accepted. So actually today, in terms of model development and how we are trying to develop assays, I'll focus on inflammatory bowel disease because we think that it's an area where these patient-derived organoids have a lot of potential to contribute on better options for patients. Until organoids were not here, it was very difficult to represent the epithelial component in the bench in order to develop drugs at preclinical level. So most of the drugs that are available to target patients with IBD come by trying to mitigate the immune response and the inflammatory response. And actually, still, there is a percentage of patients with IBD that do not respond to the available treatments. So we think that now, by bringing the epithelial component, we can bring more value to the development of drugs that works for these patients, and also to understand the complexity of the disease. So as the first activity we do when we want to address a specific therapeutic area is to develop a relevant organoid biobank. So we have already more than 10 years of experience developing these organoid biobanks in which we get access to patient material. We can get access to tissue. We can establish the organoids. The organoids are expanded in order to create what will be master and working cell banks. And then during the expansion of these models, we take into account very important QC criteria to develop models that fulfill the standard that industry expects in terms of, mycoplasma-free confirmation of the identity, growth validation, high recovery from thawing, and also basic characterization of the organoids. And then once we have these models, we can confirm that we can work with these organoids, and they are characterized and they represent, relevant characteristics of the disease. Then we go to the next step of developing relevant assays. So in the context of the IBD, we have already a biobank that consists in around 30 models from 15 different patients, in which we can represent patients that have been diagnosed both with Crohn's disease or ulcerative colitis. And actually, we have generated organoids from different regions of the intestinal tract. So in this sense, we already have a nice collection of patients in which they've been SNP validated for risk lossy for IBD, and also some transcriptomics to have an understanding of how well these organoids maintain some of the hallmarks of the disease. But once we have this collection and these organoids available already in our biobanks, as I say, we develop then different assays to address maybe a specific parts of relevant characteristics of the disease. And I just want to bring already the attention that we are able to develop from relatively simple to more complex assays, depending on the question we want to answer. So we like to take this more modular approach that depending what question we want to address, either we only have the epithelial component because part of the relevant response to the or the hallmark of the disease have to do with these only with epithelial cells. And when it's necessary to include other cell types, then we develop these co-cultures. So I'll show you now on the rest of the presentation 3 examples in which we have developed 3 different assays to address 3 different aspects of such a complex disease like it's IBD. So I start with the generation of these patient-derived monolayer assays, in which we can interrogate the activity of compounds that may have an impact on the epithelial barrier integrity that we know is one of the characteristics of the IBD patients. The fact that this epithelial barrier integrity is damaged in these patients. So we have developed, so the organoids are normally expanded as 3D structures. They grow very fast and they grow very well in these conditions, but we can also then plate them in these kind of transwells to create this monolayer that resembles and mimics the same monolayer of epithelium that our intestine consists of. And then we can, in this case, reproduce a cytokine damage assay in which we add externally some cytokines that it's known are present on the inflammatory phenotype or inflammatory environment of patients with IBD. And then then we can see that when we incubate the organoids as monolayers with different concentrations of these cytokines, and in this case is TNF and interferon gamma, we can see how the monolayer is damaged. Sometimes it's very obviously clear when looking at the surface of these monolayers. We see that the monolayer nicely generated in the control conditions is completely disrupted 24 hours after being exposed to the cytokine damage. And we can see also by measurement of tear that is this resistance that can be measured by the presence of the monolayer. We see a decrease on tear measurement when we increase the concentration of the cytokines. So in this sense, we can see how our cytokine challenge assay can induce a loss of barrier function in a dose-dependent manner. And also very nicely, we can see how this damage that again we can measure by tear measurements and correlate with barrier integrity, but we can also check viability on these monolayers, or also permeability. So we have several readouts in this monolayer assay. We can see how the damage that we were seeing when increasing the concentration of the cytokines by the drop of the green signals in tear when we are at 5 nanograms ml for this specific patient-derived organoid. We also see an impact on the viability and an impact on the permeability. We can see that also when the monolayers are exposed to tofacitinib, and this is a compound that is currently used in the clinic and for which some IBD patients have a positive response, we can see how the barrier integrity, the viability, and the permeability is restored when we are using one of the therapeutic modalities available for IBD. So in this sense, if there will be development of other type of compounds that will try to protect either barrier integrity, viability, or the permeability of the monolayer, we believe we already have a quite robust assay setup to assess the activity of these compounds. And actually, we're quite excited because indeed we have seen recent publications that are also showing that there is a correlation between organoid response to tofacitinib and clinical response on IBD patients. So another modality of assay that we have developed in order to address a specific question of the complexity of the IBD disease is the co-culture of organoids with fibroblasts. So in the context of IBD, it's very well known that there is an interplay between the epithelium, the immune cells, and the fibroblasts. And what triggers what, I think that still there are a lot of questions that need to be addressed. And we believe that, okay, now that we have the epithelial component with the organoids, we can set up co-cultures now with fibroblasts and develop assays where we can understand better this or interrogate this interplay between these two different cell types, and also evaluate activity of compounds. So in this sense, we have developed what we think is a unique platform to screen novel compounds that could target the interaction between epithelium and fibroblasts in the context of IBD. So one of the first experiments we did was to do the co-culture of the organoids and the fibroblasts in this in a 3D context, not monolayer, 3D. And actually, we can see very clearly that there is a tendency of the fibroblasts to immediately interact with the organoids, have a cell-cell contact, and actually impact in terms of the level of aggregation of these organoids. So if in these co-cultures, and here you have the measurement of different soluble factors secreted either by fibroblasts or mainly by epithelial cells, we can see that when we have fibroblasts in culture with organoids, the fibroblasts secrete a specific cytokine. But actually, we can also induce inflammatory. Oh, sorry. So this is with normal fibroblasts because this is a soluble factor for inflammatory fibroblasts. We can see that when we co-culture organoids and fibroblasts, we don't see any secretion of this cytokine. But actually, when we induce an inflammatory fibroblast by exposing these fibroblasts a specific combination of cytokines, and then we combine them with organoids, we can see that the fibroblasts are start secreting certain this soluble factor that is also, of course, present when we have inflammatory fibroblast alone. But also importantly, when we in the same co-culture experiment, we measure epithelial chemokines, then we can see that when the organoids are alone, well, we don't see any response. When the organoids are co-cultured with fibroblasts, normal fibroblasts, we also don't see any response from the epithelium. But then when the epithelial organoids are co-cultured with inflammatory fibroblasts, we can see the secretion of this epithelial chemokine, suggesting that there is a communication, a crosstalk between the inflammatory fibroblasts, and there is a response from the epithelium to this specific co-culture. And well, there is a small secretion of the same chemokine by the fibroblasts, but we can see clearly that there is an epithelial response when they are in contact with this inflammatory fibroblasts. So understanding that we could already by measuring the supernatant, we could see and detect a crosstalk between organoids and fibroblasts. We developed an assay in which we were aiming to have several more readouts just to combine maybe not only soluble mediators that we can measure by collecting the supernatant of these co-cultures, but also evaluate some agnostic organoid morphology image-based readouts to see if there is any change in the behavior of the organoids, and also some viability or death, a cell death signal from the epithelium or the organoids to be able to have also a kind of an indication of epithelial damage. So with this idea of having a 3D co-culture assay between organoids and fibroblasts in which we can have multiple measurements to understand better the crosstalk between these different cell types, we developed this InFocus platform in which we are able to already see some significant changes in some of these parameters when we have these co-cultures. So this is just a representative image of one well for each of these 4 different conditions, in which we see the PDO organoids alone, and then the organoids have still this compact structure. The organoids are just dispersed along the well. And actually, if we co-culture the organoids with cytokines that trigger inflammation in fibroblasts, so not in the epithelium, we don't see any response. So the organoids are not responding to this in the inflammatory trigger. If we combine the organoids with just fibroblasts not being stimulated, what we see, and it was a correlation with the previous image I showed to you, we see that the organoids tend to aggregate. So there is a clear interaction between organoid and fibroblasts, and this kind of stimulates the aggregation of the organoids rather than being here dispersed. So we can already see a change in the behavior of organoids in the presence of normal fibroblasts. But then when the organoids are co-cultured with fibroblasts that have been receive an inflammatory trigger, so here we have inflammatory fibroblasts, then we can see that the organoid morphology is changing. The organoids are becoming larger, more cystic. The wall is more thinner than when they are just by themselves. And actually, we are able to measure this kind of, well, we call it organoid swelling because each individual organoid increases in area from these co-culture wells, right? So we have an image-based readout now that we can measure an organoid area, and then we don't see, again, as the pictures were showing, we don't see any changes on the organoid area when we add into the organoids the trigger that induces inflammation in fibroblasts. But then when we have organoids and fibroblasts, we see a change on the organoid area, and this change is due to this kind of aggregation between different organoids. There are not that many changes at the individual level of the organoid, but then in the co-culture with inflammatory fibroblasts, then we see a significant change on the organoid morphology. And actually, we also can correlate this change with changes on cell death. So if you look at the dark green bars here, you can see that when there is no in this case, the cytokine that we are inducing here is the cytokines that we were using in the PDO monolayer to induce epithelial damage. So if we go to conditions where there is no damage in the epithelium, and then we see the viability, okay, between PDOs and fibroblasts, or PDOs with fibroblasts that are triggered with an inflammatory, that are triggered with an inflammatory cytokines. We don't see significant differences. Of course, there is a slight increase of cell death of organoids, but very minimum. However, what we think that it's also very relevant is that when we add cytokines that damage the epithelium, and this is just organoids alone, we see an increase on the cell death because the cytokines, as I say, are damaging the epithelium, and one of the responses is cell death, and we can clearly measure. And actually, when the organoids, so in this condition where the organoids are cultured with fibroblasts, and I just need to remind you that in this condition we don't have the cytokine damage for the epithelium, but still these normal fibroblasts, we see a protective effect of the fibroblasts. So when the cytokines to damage the epithelium are added, the impact on cell viability is reduced. When they are normal fibroblasts, but then when we have inflammatory fibroblasts, we lose this protective role of the organoids. And all these responses are in line of what is expected when there is an interaction between epithelial and fibroblasts in a normal environment or in an inflammatory environment. And actually, what is also quite interesting is that in this experiment, in this assay, we can also evaluate the activity of how certain compounds, and again, we are using tofacitinib as a standard of care available treatment for IBD. We can see that again, so this increase on the organoid change morphology that I was referring before can be reduced and minimized when we add tofacitinib in the co-culture, so that this protective effect that we could see for tofacitinib on the barrier integrity on the monolayer experiment is also observed on this more complex assay in which we are now evaluating the crosstalk between organoids and fibroblasts. And then the same goes with when we measure the cell viability. So again, when there is not cytokines that damage the, and I have to apologize because here the legend is swap, sorry. So in the green, dark green is the control condition. So there is not cytokine damage for the epithelium. Well, we don't see that much impact on the viability of the organoids, and actually a small, recovery from the combination with tofacitinib. But what we think it's relevant is that when we are damaging the epithelium, and we can see an increase in the cell death of the epithelium, where we see some protection where we are using normal fibroblasts, but no protection when we use inflammatory fibroblasts. As soon as in combination with inflammatory fibroblasts, we include tofacitinib, we see that there is a recovery, a protection towards the cell death response in the presence of tofacitinib. So with two different readouts, we can see the same effect, that the presence of inflammatory fibroblasts impacts the viability of the organoids, but this impact can be reduced or neutralized by the incubation of a compound used in the clinic for IBD patients, like it is tofacitinib. And actually, just to show the flexibility of the system and that we explore all the possibilities, is that in the initial monolayer experiment that I showed you before, we can also set up now monolayers in which we have fibroblasts included. So we have contact fibroblast organoid now as a monolayer rather than as a 3D structures like in the InFocus assay. And then, well, here is just an histology to show that we can identify the cell-cell interaction between fibroblasts and the intestinal epithelial cells. What we can see when we are using TEER as a measurement, so the integrity of the monolayer, we can see that when we have a normal monolayer, we can see how the TEER measurement gets built. And then when when there is a moment that we do cytokine damage, but in this condition is control and we don't do, there is not that much change on the monolayer integrity as a using TEER as a readout. Very importantly, when we add the cytokines that damage the epithelium, we see a drop on TEER. But then when we add these fibroblasts, we can see, and I think that the most relevant one is the gray compared to the pink. So this gray compared to the pink, when we have, fibroblasts and the monolayer in the presence of the cytokine damage, then we have a rescue or there is a protection with the presence of the fibroblasts to this cytokine damage that impacts the epithelium. And then, I mean, in this sense, we are also already exploring other combinations, maybe more on the context of the 3D on rather than having this external cytokine damage by the addition of the cytokines to already combine with immune cells. To have another layer of complexity to address a specific. So to have an assay that requires maybe the evaluation of compounds that are targeting towards immune cells and not only fibroblasts to see the impact on the epithelium. So with this, I will also have a couple, one or two more slides about how we also develop a swelling assay as a disease-relevant PDO-based assay, since this kind of organoid swelling has been already correlated to, kind of a proxy for IBD diarrhea phenotype. So in this sense, this is a video where we have used PG2 as an inducer for this organoid swelling, and we think that having this swelling assay with PG2 is quite relevant because it's PG2 plays a role in the inflammatory phenotype or inflammatory landscape that is observed on patients with IBD. And we have an assay in which we can see that if we increase the concentration of PG2, and these are different concentrations, we can see an increase on the organoid swelling over time compared with not having any stimulus, not adding PG2, and we can measure this also as an area under the curve. And actually, we can see that this PG2 dependent swelling, induced swelling that we see on the organoids is depending on CFTR, an ion transporter, an ion channel present on the intestinal epithelium. So in this sense, by using inhibitors of CFTR, we can see that we can again slow or reduce the swelling capacity that we can see in the organoids induced by a PG2. So in this sense, again, we have a platform in which potential compounds that could impact the ion imbalance and subsequent diarrhea that IBD patients suffer. We have now an organoid-based assay with physiologically relevant inducer for the swelling available and ready to test additional compounds. And then just to finalize the presentation, I have one or two slides also to show you how we are developing also solutions from our platform for the toxicology area. And most of them are from they are mainly focused now on the gastrointestinal toxicity. This is an area of toxicity where we believe that organoids can bring a value, especially to predict what will be the toxicity effect in human patients, since there is an historical understanding that not always the in vivo data can can predict what will be the toxicity in humans when a compound toxicity is evaluated on preclinical animal species. And therefore, we started putting some efforts to kind of validate existing compounds that have shown, well, either positive GI toxicity because they induce diarrhea, or negative GI toxicity in patients. So this is human or in vivo patient data from which, when we have performed the screens with organoids, either as organoids as a 3D structures and then viability as a readout of the toxicity of this compound, or when we set up the organoids as monolayers and then the measurement of tear and barrier integrity as the readout of the impact of these compounds, we can see consistently that those compounds that are considered no diarrhea inducing, and here I label all in pink, do not have an impact on organoid viability as 3D structures or barrier integrity as a 2D structures, and those compounds that have a diarrhea-inducing phenotype are the compounds labeled in green, in greenish or bluish. We can see that these ones are indeed showing at different levels with different ICs, IC50s, either an impact on the viability of organoids as 3D structures or an impact on the barrier integrity when we plate them as a 2D monolayers. And another aspect of our technology can support on the toxicology space is about, identifying the value of still in the gastrointestinal toxicity, the value of having animal-derived organoids from different species. So in this sense, we've put an effort to have not only organoids from a small intestine and colon from different patients, but also from different species, from one side to validate data that has been observed in vivo about different sensitivity between different species. So this is an interspecies sensitivity in which for example, SN38, it was known that is one of the compounds that I showed you in the slide before about showing diarrhea impact on patients. So that is also represented here. But it was also known that while dogs show some sensitivity and SN38, this is the active compound of irinotecan, for those that are not familiar, dogs are also sensitive to this compound and show toxicity in their intestines. Some species of rat do not show any sensitivity and any response to this compound. And now we are extending the number of compounds that we are evaluating this historical difference interspecies sensitivity with the idea to support, and the next slide is a showcase in which we can support an educated selection of the animal model if in vivo studies need to be performed, that represents better what could be the toxicity expected in humans. And in this case, we had a collaboration in which there was a compound, an anti-cancer agent, for which the toxicity studies had to be performed in vivo, but there was some doubts about, okay, what should be the in vivo model to use to perform these preclinical in vivo studies. Definitely there was the decision, rat was one of the models selected, but there was the need to select a non-rodent in vivo animal model, and in our collection we have dog and mini pig derived organoids. So we perform this viability assay in 3D organoids from the different species, and actually we could see that if we look at the response in human organoids, that was expected already some toxicity because it's an anti-cancer drug. And rat was already selected as one of the preclinical models. When comparing the response of dog and mini pig, we could see that mini pig was responding very similar to rat, while the dog organoids were responding more similar to human. So in this sense, the organoids were advising to include dog and not mini pig as the non-rodent in vivo species to perform all the toxicity studies in order to gather all the relevant data required to have information about dose escalation when moving into clinical trials, for example. So this is just an example where we are showing the capability of our platform or our solutions of the solutions of our platform in the toxicology space. So with this, I will finalize my presentation just to bring a bit this idea that we believe that by bringing patient-derived organoids, we are having a more patient-centric solution to do drug development. We are convinced that the earlier organoids are introduced in the pipeline of drug development already to assess some target identification, biomarkers, identification of hits. We believe that then the time and the money spent to develop a drug that moves into the clinical phase will be significantly reduced. And this is because the organoids have patient predictive value. They represent a more human relevant characteristics in terms of antigens, for example, than cell lines or animal models. And with the flexibility of the technology, to develop as many assays, as many readouts are required to address the specific question, we believe that our platform is a solution to support your drug development pipeline. With this, I finalize my presentation, and I'll be looking forward to receive the questions from the audience and have some of the discussion. So thank you very much for your time, and looking forward for the discussion.
Thank you, Sylvia. Before we go over to questions, I'd like to remind everyone that this webinar will be available to watch on demand at ddw-online.com. Sylvia, if you're ready, if you'd like to turn on your webcam, we'll go over to questions. Hi.
Yes. Hi.
Okay, so our first question is about PDCOs from biopsies. The question is there any specific protocol or tricks or troubleshooting to increase the efficiencies?
I will assume that when they mean PDCOs biopsies, they are talking about establishing from cancer or from tumor biopsies. So with this assumption, I'll say that one of the important tricks is to try to process the sample to establish organoids as soon as possible. So because the amount of cellular material, tumor cells available in this biopsy to establish the organoids is limited, and the viability of these cells can be impacted by the time waiting to process the tissue. We have seen a significant increase in efficiency if the biopsy is processed immediately for model establishment. Another important one is at least when seeding the isolation or the material that has been processed, to try to seed it in the minimum volume of matrigel as possible, so not more than 2 wells of a 24 well plate. Also to try to ensure that the limited number of tumor cells available in the biopsy are together, and we believe that a part of the media that we are providing, there is an important paracrine secretion from these tumor cells. So the more concentrated we have the tumor cells from the biopsies in a well, it also increases the efficiency to establish an organoid culture out of this biopsy.
Okay, that's great, thank you. Our next question, I'm trying to make patient-derived breast cancer organoids. I'm using the standard protocol, which is generally used in literature. However, we get less cells after the dissociation step. Can you help me to improve, and how can I get more cells?
Well, it's a bit difficult to know exactly, because I guess there are different publications. If the person feels that it's missing, I don't know, the amount of cells they have at the end of the process to establish an organoid compared to the initial material, I think that will be important to consider maybe the centrifugation steps or the washes that, I don't know, if the cells do not go down, that they can be lost. I think it's also important to emphasize that for an organoid establishment, there is not need to reach a single cell step level. if the tumor cells are aggregated and make, they're almost the organoids are there because these large structures, these are more efficient to establish a culture. So yeah, it's a bit difficult to address very specific technical questions. So maybe I invite this person to provide a bit more detail on exactly what protocol is using that I can maybe address after this webinar to provide more useful tips to help about this.
Okay, yep, no problem. We'll follow up on that one. In Biobank, how many passages of PDCO are stored, and after how many passages of chemotherapy does the response change?
Well, so in principle, if the organoids are passaged and are cultured properly, we have not seen a change on the chemotherapy response. So actually, this is the reason why we create these biobanks, and we cryopreserve the organoids at different steps of passage level. So from isolation, and of course, it depends a bit about if it's a normal or a disease. If it's from a type of cancer where organoids proliferate a bit faster, for example, pancreatic or colorectal, than maybe breast or ovarian, where the organoids grow slower. But we normally establish what we call the master cell bank after 5-6 passages. This is what normally you need. Again, it also depends on your starting material, but as an average, between 5-6 passages, you can make already a full plate of a 6-well plate, and this is what we cryopreserve as an MCB. Then we thought this vial expanded 4 or 5 more passages, and then freeze another set of models so we can multiply better and preserve the specific patient longer. And then when we perform our experiments, either to assess their activity, to their response to certain compounds, and maybe use this model repetitive in different projects, using different combination of compounds, but always having a couple of control compounds that are always evaluated. We don't expand the organoids more than these 5-6 passages you need to have sufficient material to execute the experiment. And following this procedure, you don't see a change to the response to chemotherapies. So normally you can expect, if you observe a change in the response of the organoids over passage, it means that during this passaging, either the organoids were not processed properly or were not refreshed on time, or something happened with the media or in the incubator that, of course, if there is a kind of a selection, the organoids are going to adapt. And maybe if we talk about cancer models, the clonal heterogeneity may be lost, and therefore you start seeing changes on the response. But if these patient-derived organoids are processed properly and with the right reagents, it's not expected to have a change in the response of compounds.
That's great, thank you. For the experiments with monolayers, are the cytokines added in the apical or basolateral compartment?
So we have tried all the combinations. But normally we do it basolateral because this is the natural way that the epithelial cells will get exposed to these cytokines.
Great. Next question. How do you control the relative growth of tumor versus fibroblasts to prevent the fibroblasts outgrowing the cancer cells?
So this problem, you normally have it when you need to process tissue and do the isolation. It's true that on the establishment of an organoid culture from the tissue isolation, sometimes you bring into the cell preparation a fibroblast. Normally, what we see is that these fibroblasts tend to attach to the base of the plate. So you can see that in the matrigel dome, the organoids are growing inside the dome, but then you have some cells growing as 2D as a 2D layer. These are mainly the fibroblasts. If at the moment of collect the domes for the next passage, you don't scratch the surface of the plate very carefully, very gently. You try to just aspirate the domes. Then normally the fibroblasts stay on the old plate, and then you collect your domes, transfer into tube, do all the mechanical dissociation to disrupt the organoids, and seed them in a new matrigel solution to sit in a new plate. Then by this process, after the second, third passage, we don't see the fibroblasts growing on the culture where we have the organoids.
Great, okay. Our next question, I imagine there's quite a lot that can be said on this, but you might have to just give an overview. So the question is, can you elaborate on the advantages of organoids in comparison to primary cells and co-cultures?
So the difference with primary cells is that normally in the current situation, the primary cells directly from patients are available to perform one experiment, and that's all. With the organoids, we believe that they are kind of primary cells that have the capacity to be expanded and cryopreserved, and this allows that the experiment and the data that you generate for the first time, you can reproduce and repeat in further, over time in the same over different passages. So in this sense, if you think about the development of a compound for which you are doing some molecule development. With primary cells, you are always testing one time on different patients where you can have different responses because patients are heterogeneous and they have an heterogeneous response. So with the organoids, we can represent all these patients and have these patients available along all the process of drug development, to keep developing and comparing, from going from hit to lead and developing your compound further. The comparison about the co-cultures, I'm not sure if I'm going to understand the right perspective. If not, this person can make a comment. If the co-cultures means, I don't know exactly what it means, but I will assume that if it's about not primary cells, but tissues, so you get a biopsy of a patient where you only don't have the organ or the tumor cells, but also other cell types like immune cells or fibroblast or endothelial cells that are present there. I think that in this sense, you have a bit more complex media, but the point that you can only perform this experiment once, and then either it worked or did not work, but how reproducible is this result becomes a bit more challenging because of the difficulty of having the same patient available to perform more than just one experiment.
Brilliant, thank you. Do you think the organoids fibroblast co-culture model fits better for evaluating targets in epithelial cells or fibroblast?
Well, I think that of course it is a platform that allows to test any compound that it's aimed to integrate the crosstalk between the epithelium and the fibroblast, right? So I think that of course there is a lot of understanding that the fibroblast creates secrete cytokines and make some matrix remodelation that can impact the epithelium. But at the same time, we also are seeing on these co-cultures that the epithelium also to the response of the presence of the fibroblast secretes certain cytokines and certain products that at the same time have an impact on the behavior of the fibroblast. So we believe that this assay can address any kind of compound that impacts the crosstalk between these two cellular types.
Okay, thanks. Our next question says, great presentation. Do you have any tips to live cell the organoids for long hours?
Sorry, a tip about?
To live cell the organoids for long hours? I'm not sure.
Yeah, I really don't
Maybe
understand this
follow up
question.
on that one. We're not sure on
Yeah,
that one.
maybe if he can provide a bit more detail, yeah, I can address. Sorry about that.
No problem. Okay, the next one. How long does it take to produce organoids?
Very high level, because this depends about if you get a biopsy or a resection, because the amount of material is different. And something that we have also experienced is that when you try to establish a normal or not cancer model, actually the organoids grow much faster than a tumor model. We will say that, just to give two examples in this context, when we make organoids from a biopsy from patients with cystic fibrosis, where they have a monogenetic disease, we can make a full plate of a 6-well plate, and this is normally what we freeze in our MCV level. We can do this in 2-3 weeks. There is enough time to expand, to establish the organoid culture and expand it to a full plate. when you do a resection of colorectal cancer or pancreatic cancer, you can get this full plate in around 5-7 weeks. And if you will try this from a biopsy of, for example, colorectal metastasis in liver, you can make this full plate in around 10 weeks, 12 weeks. Very high level. So it really depends on the amount of the starting material, the more the better, how fast the models grow. And we see that in some cases, normal grows faster than tumor models, and what type of tissue it is, because we also see a different proliferative capacity for example, intestinal compared to breast, or ovarian, or head and neck.
Okay, great. I'm sure that will be really helpful. Our next question is, do you have experience with ovarian cancer organoids? Are you able to establish organoids from every sample, or are there some that do not form organoids?
No, so we have seen a different level of efficiency establishment of organoids. If you exclude, again, the quality of the sample that you receive, because if the tissue is very necrotic, or the tumor content is very minimum, then you will not establish the organoids. And I think this has nothing to do with the organoid technology, but with the quality of the material you receive to establish such models. But still, we see a difference in where, as always, intestinal derived models, we are very close to a 99% efficiency for normal tissue. Then it drops to maybe around 85-80% for cancer models from the intestinal side. And in our hands, ovarian is around 50-60% of the model. So in this sense, we are not at the same level than intestinal, and we are still trying to understand if there is any space to improve the media, if there is something that maybe on the initial formulation can be improved to increase this efficiency. If it's a bit more about the nature of these tumor cells that having less proliferative capacity, it could be that then if you don't have sufficient material available, it becomes a bit challenging to establish a culture that can be not only characterized but used for further experiments. I think that, in principle, from all samples, again, if you have sufficient tumor cells, you can establish a tumor organoid culture. But if this grows fast enough and allows to generate enough material to then cryopreserve and do further experiments, then for ovarian, the efficiency is lower than other tissues.
Thank you. The next question relates to intestinal tissues. It says, is it feasible to generate PDOs from severely inflamed intestinal tissues, as this is a known cause of stem cell depletion?
So from our collection of the IBD biobank, we were able to establish models from regions that the radiologists identify as an inflamed regions. Of course, it's very important that the biopsy is able to collect part of the crypt where even if they are depleted, there are still some stem cells. We have not seen a significant difference in efficiency when we establish our IBD biobank, if the sample was coming from an inflamed or inflamed area. So in this sense, I think that the quality of the biopsy to secure that you get the crypt of the intestine represented on this biopsy to succeed establishing the model, even if it's from an inflamed region, is a critical one.
Thank you. I've got a couple more questions if you're happy to carry on. We've gone a little bit over time.
Sure.
So organoids are very hard to transfect. What is the best choice for this application, polymer or lipid-based reagents?
Well, our best experience, I guess that it depends for what purpose you want to transfect the organoids. We see that, for example, in our hands, electroporation works better than using any kind of, lipofectamine or similar products to transfect. Still, the numbers are relatively low in terms of percentage of positive clones, depending what you want to do. Another one that also works quite well, but maybe has for a very specific applications, is the use of lentiviruses. In this sense, the organoids are much more efficiently, at least you can introduce much more efficiently exogenous DNAs. So yeah, at least this is our experience in our hands. We don't introduce exogenous DNA to organoids using these lipofectamine-based approaches. We use electroporation.
Thank you. The next question is, have you compared the efficiency of organoid generation depending on the matrix, so whether it's matrigel, Geltrex, etc.?
We've done a very high-level comparison. I mean, in principle, all the animal-derived ECMs are good to establish models. There is only a very slightly seems that there is a slightly preference or slightly better numbers, but very slightly when using in positive when using matrigel, especially for the normal tissues.
Okay, thanks. The next question, what is the source of the fibroblast used in the co-culture? Are the fibroblasts from the same patient as the organoids?
So when we started this project, we first just got access to fibroblasts, patient-derived fibroblasts that you can purchase in these, cellular providers, just to optimize the assays. When we saw that, that this co-culture assay was working, we started also the process and developed the protocols where we can isolate the fibroblasts from the same patient. So the data that I've shown you here is from a fibroblast primary line provided, from tissue provided by a cellular provider. But now we are developing this. We are isolating these fibroblasts from the same tissue from which we established the organoids, and we have not tested yet. But then we will try to run the experiment also now with the same, let's say, autologous system for fibroblasts and organoid.
Okay, thank you. In the slide you showed, inflammation is induced by cytokine. Have you tried culturing IBD organoids with fibroblast?
Yes, I mean the organoids that were used on this in-focus co-culture and are IBD-derived organoids. So just to make it clear, we add two types of cytokines. One of them are to induce the inflammatory phenotype into the fibroblasts, and then have the IBD organoids co-culture with either normal fibroblasts or inflammatory, inflamed, induced fibroblasts. And then in other cases, when we use cytokines, is to induce the damage on the monolayer or in the epithelium. And in this case, these cytokines are replacing the absence of immune cells in these co-culture experiments.
Thank you. I think this better be our last question for today. So after dissociation of tumour, do you use strainer to remove stroma in PDCO formation procedure?
Yes, I think that no, we were not doing this at the beginning, but now in our isolations after the digestion with collagenase type enzymes, we filter the solution just to make sure that we get rid of big chunks but I think it's more about getting rid of big chunks that can impact. I think that they can impact the volume in which you need to seed the organoids. I think that one of the initial questions was talking about tricks, and we believe that especially when you work with a sample that may have limited tumor cellularity, the more you can concentrate these tumor cells in one single well, that this paracrine secretion that the tumor cells have and supports for the organoid establishment and growth, can be impacted if you have a very rich pellet, because you have just a lot of material that is not relevant because it's not has the tumor cellular content, and in this sense we will do the filtering to get these big chunks out and generate a very small pellet that we can concentrate in a large in a matrigel drop in one single well to increase the efficiency of establishing the tumor organoids.
Thank you. Thank you for your excellent presentation and for taking the time for this discussion. It's been really good.
Thanks to the audience for the interesting questions. So it was a pleasure.
Brilliant. Thank you so much. I'd just like to remind everyone that you can listen to this webinar again on demand at ddw-online.com, and you can view Drug Discovery World content online or on your mobile or tablet. Thank you for listening, and thank you, Sylvia.
Yeah, thank you.